Hi, my name is Maureen Sampson, and I'm the student representative for the Molecular Toxicology Interdepartmental Program. And we are so excited today to host the lovely Dr. Andrea Gore. We've actually been trying to get Dr. Gore to our campus for a couple of years now, so we're so excited to have her today, not only on our behalf, but this event is also co-sponsored by the Center for Study of Women, CSW. Dr. Gore is the Vasek Chair of Pharmacology at the University of Texas, Austin, and she's also the esteemed editor-in-chief of Endocrinology, a wonderful journal. Dr. Gore received her PhD from the University of Wisconsin-Madison in 1990. She did a short stint, well, I guess it wasn't short, but she was at Mount Sinai for a few years and then has been at the University of Texas, Austin. And she did her undergraduate work getting a degree in biology from Princeton. Dr. Gore has hundreds of papers that have been cited nearly 5,000 times, according to Google Scholar, so she's had a very big impact in her field. And as you guys know, with the nature of our modern world, the diversity and burden of chemicals in our environment that we're exposed to is increasing every year. And an important aspect of these exposures for us to consider is the effect that they have on our development, of course. So Dr. Gore studies how endocrine disrupting compounds, so like estrogen mimickers, affect the development of the brain, and also looking at aging and menopause, how these chemicals also affect the aging process. And she also looks at the dimorphism between how these different chemical exposures affect men and women. So we're so excited to have you. Looking forward to your talk.
Thank you. Thank you for the kind introduction and the persistence in the invitation. I promise I was not playing hard to get. It was just trying to coordinate the teaching and other travel and so on and so forth. But I'm really glad that I was able to be here and to meet with all of you. I'm really sorry about the color of my slides, which look nothing like this in real life. So just bear with me. I hope that you'll be able to see most of the important things like the data. But if the colors don't quite look perfect, just please forgive me for that. Okay, so let me start by saying something that is incredibly depressing, which is that we have irreversibly contaminated our world. And just as an example of parts of the world where there actually has been no industrial activity and where chemicals have not really been a part of people's lives, for example, in the Arctic or the Antarctic, concentrations of industrial chemicals are found in the bodies of these people and in marine mammals and in other animals that they eat. They're in the food chain. And that's because there are so many tens of thousands of chemicals in the environment. Many of them can get into the air currents or into the water currents. They get into the food chain of species that might feed in a contaminated area and then spend part of the year up in the Arctic where people are eating these contaminated foods. And this gives you just a sense of what synthetic chemical production has looked like from 1900, beginning really in the mid-1940s when we had what we consider to be the beginning of the chemical revolution, which really started during World War II when countries were trying to develop chemicals to kill people from other countries. But we also began to appreciate the importance of these chemicals in day-to-day lives. And so here's the curve through the mid-1990s, and this has continued upwards over the last 15 years as well. So not every chemical out there is what I define as an endocrine disrupting chemical, or EDC. The definition I like to use for an EDC is an exogenous chemical or a mixture of chemicals that interferes with any aspect of hormone action. So this was from a consensus group at the Endocrine Society that wrote what we call a statement of principles, and I like to work off of this definition. There are many different classes of EDCs. So here are just a few examples of industrial chemicals, household chemicals, personal care products, even heavy metals in some cases can act as endocrine disruptors through interacting with hormone systems. So let me first try to convince you that EDCs are relevant to humans. So work being done all over the world, but there's been really a heroic effort done at the Centers for Disease Control, the CDC, where they have these national surveys where they've been monitoring people's health, asking them questions, and also taking blood or serum or cord samples, and then using those samples to measure what chemicals people might be exposed to. And you can see what some of these statistics are, that in human cord blood, 287 different chemicals have been found in umbilical cord blood, which will reach the fetus. Now, not every person has 287 chemicals in their cord blood, but across people, there have been this many chemicals found. They've also been found in breast milk when babies are suckling. And of people whose urine or serum or fat samples have been tested, you can see what the percentages are of these people who have detectable levels of bisphenol A, which is in plastics, phthalates, which is also a plasticizer, other kinds of compounds. The chemical that I study in my lab, polychlorinated biphenyls, or PCBs, let me see, 100% of people have PCBs in their body. Incidentally, these chemicals were banned more than 40 years ago, and if a baby is born today, she will have PCBs in her body because it's so entrenched in the food chain. It's in every single person. So, yes, very depressing. So I'm going to segue very quickly into my research because I know that this is kind of a short class, so I want to be able to get through everything. But if you have any questions, please don't hesitate to interrupt me or ask for any kinds of clarifications. My interest is understanding the effects of EDCs on the developing brain. And let me explain to you what my motivation is for doing these kinds of studies, because we don't usually think about the brain as having much to do with the endocrine system. But the brain and the endocrine system communicate with one another through the brain's neuroendocrine systems. And that's the system that I've been studying in my lab and which Maureen was talking about at the beginning. And I'll get back to that in a moment. The brain has some of the highest concentrations of receptors for those hormones that we know are perturbed by EDCs. So it's incredibly responsive to both endogenous hormones as well as compounds that may be exogenous to the body but may activate or inactivate hormone receptors. We also know that there are developmental sex differences in the brain that begin in the fetus and continue into infancy and even into childhood, because male and female fetuses and babies have different gonads, and those gonads make different levels of steroid hormones. They make the same steroid hormones, but males have much higher testosterone, females have much higher estrogens. And those differences in exposures to hormones in our body, but also to our brain, result in a sexually differentiated brain. I study the hypothalamic pituitary gonadal, HPG, control of reproduction. We also know that reproduction is a target of endocrine disruptors, in part through their actions on the brain as well as on the gonad. And I'm going to talk about some of the sex differences and why they're important to this field in terms of development, brain function, and also behavior. So here's sort of a snapshot of the general model that we're studying and that we happen to think is relevant to humans as well as to rats. Of course, we do the research in my lab on rats. But what I'm interested in general is exposures to natural hormones, or EDCs. Typically, we do our exposures to pregnant dam rats, which is a critical developmental period for both hormones in the brain. And then we follow the offspring, what we call the F1 generation of rats, as they're born, as they are infants and become little people and grow up through puberty and become adult rats and even go into aging. And what are the consequences of these previous EDC exposures limited entirely to this prenatal period. I'll also talk, if there's time at the end, I'll talk very briefly about the evidence that there may also be transgenerational effects of EDCs from the descendants of these exposed fetuses 2 or 3 or even 4 generations later. Okay, so just to really hammer home the importance of the brain, I also want to bring you back to this picture that I showed you of the synthetic chemical production over the last 50 years or so, just looking at this curve. And the reason I'm showing you this, I want to give you some human perspective, is that certain neurological, neurodevelopmental, and behavioral disorders have been increasing in frequency over the last 50 years, some very much in parallel with these curves of synthetic chemical production. Now, I'm not trying to say that neurological disorders are being caused by endocrine disrupting chemicals. What I think is going on is we're better at diagnosing them. We know that despite better diagnosis, there's still an increase in the prevalence of these different disorders. This one is autism spectrum disorder, numbers of people. This one is bipolar disorder from 1994 to about 10 years ago. And this curve is the curve in youths who are first diagnosed with bipolar disorder. You can see that trajectory is also very similar. So EDCs are not causing these processes. However, we believe that they're contributing to this increase in many of these disorders due to their actions on the brain. Something else that's really important about these disorders are they're sexually dimorphic. Males have much higher incidence of autism spectrum disorder than females, and some people think that they may be more vulnerable to those types of actions of EDCs in the fetus. And almost all affective disorders are also sexually dimorphic. Typically, depression, major depression, and anxiety is greater in females, and so on and so forth. Okay, so any questions before I start to talk a little bit more about my research? Okay. So my lab is interested in the neurobiology of reproduction, and I want to very briefly introduce you to the system that we study, the hypothalamic pituitary gonadal axis, or HPG axis. And I wanted to orient you to it first in a typical half woman, half man that you might meet on the street of California. We don't have them in Texas, but you know. So the hypothalamus is at the base of the brain, and it has, and I'm going to show it here, kind of blown up here bigger. It has neurons, just like the rest of the brain, and synapses. But for its neuroendocrine function, hypothalamic neurons release their neurotransmitters into a blood system that vascularizes the anterior pituitary gland, which subsequently secretes another set of hormones that then go into peripheral circulation. So to look at it in the context of a person, the hypothalamus is here, and the pituitary is here right underneath it. And so then these pituitary hormones get into general circulation. So all these arrows are indicative of the direction of the hormone secretion. The target of the pituitary hormones in this case are the gonads, so the ovary or the testis, again showed in perspective down here. And the gonads release gonadal steroid hormones and protein hormones that then circulate through the body. And some of these arrows are then meant to indicate that some of the hormones also get back into the brain, they get back to the pituitary gland, and so there's this feedback regulatory loop that happens when brain, pituitary, and gonadal hormones are released. And you can also see that arrow here. Now, what we're really interested in is how these processes may be disrupted by EDCs, both the feed-forward and the feedback processes. And the feedback is particularly important in the nervous system because the hormones, you can see here, they can act upon hormone receptors within the nervous system. We know that EDCs can also act upon those hormone receptors. And I'm showing it to you here for the hypothalamus, but I also want to emphasize that almost all parts of the brain have hormone receptors at differing levels, and maybe with some heterogeneity. But other parts of the brain are also exposed to hormones and need those hormone exposures to develop normally and can be disrupted by the EDCs too. Okay. So the premise of the work that we're doing in the lab, our central hypothesis, is that exposures to EDCs reprogram these reproductive neuroendocrine circuits in the hypothalamus. And the sub-hypothesis, which I'll get to a little bit later, is that this reprogramming, not only does it change expression, how neurons are born, how they hook up, their neurotransmitters, et cetera, but it actually has phenotypic outcomes that relate to neurobehavioral or neuroendocrine types of functions. So this hypothalamic circuit that I study is very complicated. It consists of many neurotransmitters and neuropeptides, hormone receptors, other signaling molecules, et cetera. And so these neurons that I'm showing here, I'm not labeling them, but they would consist of these different types of, they would express these different kinds of molecules. Okay, so I'm going to spend just a little bit of time talking about some of the work that we've done on EDCs in the developing hypothalamus. Most of this is published, or maybe even all of it's published now, so I'm going to only show you maybe four or five slides, because I actually want to spend most of the time talking about behavior. So I mentioned that the EDC that I study in the lab are a industrial compound called PCBs. So I actually pulled this picture out of the New York Times within the last year. And the reason that this picture caught my attention was because it's showing that this is dredging of the Hudson River in upstate New York, because back in the 30s, 40s, 50s, really through the late 1970s, PCBs were used commonly in industry, and they very heavily contaminated the Hudson River upstream. Now, the Hudson River, when it goes downstream, goes to New York City, so they expose an absolutely unbelievably large number of people throughout New York State, and it's actually quite a bit of it concentrated in New York City. Now, I was at Mount Sinai School of Medicine at the time in New York City, and had met some people in community medicine who were really concerned that there were people who were fishing in the Hudson River and were eating the fish, and these people had really high body burdens or concentrations of PCBs in their body. And so I got interested in this because nobody knew what the consequences were for developmental exposure to PCBs at the time that I started my project. So it was interesting to me that they're still dredging the Hudson River in 2015 or 2016, and we still have these exposures. Okay, so let me show you just a couple of chemical structures quickly. This is a very sketchy schematic of what the natural estrogen hormone estradiol, 17-beta estradiol, would look like. It's a 4-ring structure. It has a phenol ring on what we call the A ring over here. This is what PCBs look like. It's a biphenolic compound. It's chlorinated rather than being hydroxylated, although they can also they can have a mix of hydroxyls and chlorides as well. But it's also got this structural piece that seems to enable it to interact with estrogen receptors. It doesn't interact with nearly the affinity as natural estradiol does, but it's considered to be a weakly estrogenic type of compound. And so we know that estrogens as well as androgens are important in brain development and sexual differentiation. So that was also part of the motivation for looking at PCBs in my experiments. Okay, so here's the developmental timeline now in a little bit more detail. So the way we do the work is we breed animals in our lab. They're just normal Sprague Dawley, they're outbred rats. Bring them into the lab, breed them, and on 2 days of gestation, so gestation in these rats is about 22 days. On day 16 and on day 18 of gestation, we expose them to one of the treatments. They either get vehicle, which is a DMSO in oil compound, and they get it on both of those days, or they get the PCB mix, which is called Aerochlor 1221 or A1221. We gave 2 different, either 0.5 or 1 mg per kg, which are considered to be low-dose exposures, probably pretty comparable to human exposures, only given twice. And we also use estradiol at a low concentration as an estrogenic positive control, so we can compare the effects of PCBs to estradiol and then see whether what we're seeing is similar to or different from estrogen, because that might tell us something about the mechanism. That is the only exposure the animals get. It's not toxic to the animals. We have normal pregnancies, normal sex ratios. We allow the litters to be delivered, so here's our newborns. And then we allow males and females to grow up, and then we phenotype them at different ages, and we monitor their development. And the ages that we pick are important developmental points in these animals' lives. So sort of P15 is early childhood, P30 is mid-puberty, P45 is late puberty to early adulthood, and then P90 are adult animals. Okay, so like I said, I'm just going to summarize the research that we've done looking at what's going on within the brain in terms of gene expression and protein expression. But I did want to share with you sort of a general big picture of what we're finding. So what we found, and this is probably not terribly surprising, but we found sex-dependent, age-dependent, and region-specific effects of these EDC exposures on expression of many of the genes that we looked at. And the families that were most affected were those genes. We used some what we call low-density PCR arrays. So we picked a whole assortment of genes to look at, and the ones that came up as most commonly affected by the EDC treatments were those involved in hormone responses such as steroid hormone receptors, neurotransmission, and neuroendocrine function. I've cherry-picked a list of genes that I think are really important, that I know are important for the neuroendocrine control of reproduction, but if anybody's interested in other endpoints, they're published actually in several different papers. I'll just point out that these are the estrogen receptor alpha and beta, the progesterone receptor, androgen receptor, and neuropeptides, gonadotropin-releasing hormone, kisspeptin, and its receptor, that are critical for the hypothalamic control of reproduction. If you disrupt GnRH or kisspeptin signaling in the hypothalamus, you actually will not have any kind of reproductive function at all. So they're sort of the final common path in the brain's control of reproduction. So we looked at gene expression, but it's also important to know what's going on at the level of the proteins. And I think this is important in all tissues, but it's particularly important in the brain because the brain is so incredibly heterogeneous. When you think about just taking a snapshot or a slice from the brain and looking at the expression of different neurotransmitters or peptides or hormone receptors, it's not just how many there are, it's also what is their interrelationship with one another. Are they making connections to each other, synapses? Are they co-expressing things? And so we've done a little bit of work looking at protein expression of just a few molecules to start with, estrogen receptor alpha and beta, kisspeptin and GnRH, which I told you are so important for reproduction. So I'm going to summarize these all in one slide, but we've published these as well, and I hope you can see it. It's really strange coloring. I could spend a lot of time explaining these panels, but I'll just explain these two up here so you kind of get an idea of how we do this work. So for these studies, we take our animals that were exposed to the vehicles or to the treatments. We take their brains out in adulthood and fix them, and then section them and do immunohistochemistry in specific hypothalamic subregions that we think are important for the functions that we're studying. And so we're looking, in this case, in part of the preoptic area of the hypothalamus. Now, we know in normal animals that are unmanipulated, the expression of estrogen receptor alpha is higher in females than in males. And you can hopefully actually see that here. Here's a control female. ER alpha are the black dots. It's a nuclear receptor, so each of those are nuclei. And then on the right is a male brain. You can see there are probably less than 50% of the ER alpha receptors in a male brain. And when we compared those brains to animals that had been treated with the endocrine disruptors, what we found was that the female brain estrogen receptor actually looked just like the male brain. It was very profoundly downregulated and looked actually quite a bit more male-like. So that's summarized here. The EDCs decreased ER alpha in the females. We also saw that EDCs decreased expression of ER beta. We only looked at females in that case. We looked at kisspeptin, where the expression in females, maybe you can see here it is in females, lots of little green dots. In males, you don't really see very many green dots. Much greater, like 10 times greater in females than males. They're also quite profoundly downregulated in the EDC animals. And we also looked at the activation of GnRH neurons and that are necessary for ovulation in females, and that was also downregulated by the EDCs. So we have some good protein targets. Yes?
Dr. Gore, were you able to look at all at different estimates and whether the expression would be, the difference would be increased or decreased?
Right. Right. So the question is, did we look across the estrous cycle? So not in this study. In this study, we actually looked at all the females on the day that we predicted they would ovulate. So we would have them all, it would be an apples-to-apples comparison between the different treatment groups. We've done a little bit of work looking at animals across the estrous cycle in other experiments, and we do see that it's hard to, we've looked at hormone levels, and the natural fluctuation of hormones that occur across estrous cycles is affected by the EDCs, and it's actually most profoundly affected on the day of ovulation. So I think we kind of, in a way, lucked into the day when we probably would have had the greatest effect. But the natural hormone environment will also influence how the outcomes of these studies as well, and so it does need to be controlled, but we do control it. That's a good question. Okay, so I'm going to summarize this very short section by telling you that we've discovered, and other people have also discovered, that networks of genes and proteins that are involved in the neuroendocrine control of reproduction. Other neuroendocrine processes are, we call it reprogrammed by the EDCs. The gene expression and the protein expression change, and those changes in many cases manifest throughout the life of the animals. Okay, but to me, having a gene or a protein expression effect isn't terribly meaningful if it doesn't have any sort of physiological meaning. So, of course, we were interested in knowing what were the behavioral and the physiological consequences of these prenatal exposures. Could we relate them to some of the gene and protein changes that we saw to really get at what does it mean to be endocrine disrupted? And so here's just a little assortment of some of the behaviors that we've run our experimental animals through. And I'm going to give you some little bit of data to show you some of the outcomes that we've looked at. This was actually the first study we did. It's getting to be an old study now, but I still love to talk about this study because I have a great video. And what we did here, and the reason why I actually really like this study is because people who are doing work on the effects of EDCs and reproduction usually evaluate reproduction by taking a female and a male rat or mouse or C. elegans or whatever, putting them together, and then coming back the next day and seeing, did they mate? Is there sperm in the vagina? Or, does the female get pregnant and carry the pregnancy to term? And that's fine. But the world is much more complicated than that. In the wild, animals have to make decisions about mating. There's mate choice. There's a much more complex environment. It's not just you're in the cage together so you can mate. It's there are other things, predators you have to eat, food availability. So probably about 20 years ago, people started doing what they call paced mating trials. And this really actually gets at the female side of mating. I'm a little female-centric, I have to admit. And what this does, it allows the female rat to set the pace of the mating. She can determine if she wants to mate, if she doesn't want to mate, how much time she's going to spend with the male. And it's incredibly low tech. This experiment, this setup cost me $20. I had an aquarium already in the room. We built a plexiglass partition. It has 2 holes cut out in the bottom, and the holes are small enough. The female can run back and forth. That's the female. But the male's too big to run back and forth. So he's stuck here, and she decides if she's going to go back and forth. And when we started it, we actually only had 1 hole, but she was never able to get through because his head was always blocking the hole. So we had to cut 2 holes. So what I'm going to show you now is the video. We're still doing pace mating, so I'll have to make some new videos because it's a terrible video. But what it's going to be focused on is the male side of the chamber because you'll see the female coming into the male and he'll be doing his little male things to her. You'll see her show that she's receptive. She goes into what is called lordosis, where she arches her back. and that shows that she's sexually receptive, and then he tries to mount or intromit or ejaculate. And I want you to notice that every time he does something, she runs away from him and tries to get back to what we call the escape chamber. And that's how the females pace their mating. And then what we can measure is how much time she spends, how long it takes her to do certain things. So hopefully this will work. Okay, good. And you can see how old this was. So we're doing more of this type of work in the lab, and I'll make some new videos. Okay, so they're meeting. He mounts her. Okay, she's trying to get away. She was starting to go through the hole when the... Okay, so there he is blocking the hole, but... Okay, there she goes. Run away. They are pretty fast. Okay, this is the last one. He ejaculates. You have to watch his hands. And then he, actually, it used to be a little bit longer. He kind of collapses after that. Okay. So I'm only going to show you two graphs, but they illustrate two things. And these are just examples of the kind of data. We only had one, this is one dose of the EDC, and these are the vehicle animals on the left. So if we look at the amount of time it takes the female to return to the male after he either mounts or intromits or ejaculates, she returns in about 2 minutes if she's a control DMSO animal, and she returns in 2.5, 3 minutes if she was treated with EDCs. So it takes, about 30–40% longer for her to return. And we see that with the other behaviors as well. The female just takes longer to go back to the male. We also saw that even though we were always using the females on days when we knew they were receptive, almost all the control animals mate on the first trial, but fewer of the EDC animals mated on the second trial. So you may be wondering why I'm so excited about these differences, and it really has to do with trying to put this into the picture of being in the wild, where mating is not done in a nice, safe place, where you have to be able to mate efficiently, to be able to get away, to be able to make your choices quickly, and that all seems to be slowing down in our EDC animals. I'm also going to take you through some other behaviors that we're doing. Some of these are not published yet. One of the behaviors that I'm kind of excited about are ultrasonic vocalizations of the rats, or USVs. And these are ways that rats communicate with each other, and they can communicate both positive as well as negative types of affective states. So under certain circumstances, when they're very stressed, they emit certain types of USVs. When they're very excited or motivated, and so motivated behaviors like food and sex, but also people use these kinds of calls also for tests of drug addiction, because animals that really love cocaine will also emit certain types of USVs. And so we decided to use this test in our animals to see whether maybe the way they're communicating with each other has been affected by this prior exposure to the EDCs. So let me talk about how this test works. It's a little bit complicated, and my graduate student Victoria Topper did this study. The first day we just habituate them to a sound attenuated chamber with a microphone, and we record their baseline USVs, and they don't call a whole lot on baseline. We do it again, 2 days of habituation, and then on the third day we want to evoke the USVs with a stimulus, and so the stimulus is an opposite sex animal. So if the EDC animal was a female, we expose her to a male. If the EDC animal was a male, we expose him to and the animals they're exposed to were untreated. We expose him to a female, and other people have shown that if you introduce opposite sex animals, they call to each other. But the problem is we can't record them while they're calling to each other because we can't differentiate their calls. But what's been shown is if you leave them together for a little while across this grid and then remove the stimulus animal, the animal that's left behind calls a lot. And we think that's sort of the come back to me, you're very interesting, I want to get to meet you kind of call, a positive affect kind of call, which actually is confirmed by the wavelengths of the calls that we can record from these animals. They're in what we consider to be a positive affect range of ultrasonic frequency. And I'm going to show you just a little bit of data. I actually have an audio, but I have a feeling it's not going to work. Let me see. No. Okay, well, there's different kinds of calls. And they are what they sound like. A rise call would be going up. Sorry, not very good, but a trill call is and then flat is So we think flat, and we and other people think flat calls are given just sort of general, hi, I'm here, not strong positive or negative type of affect. But the more complicated trill and rise calls tend to be more what we call hedonic calls. And so I'm just going to show you the data for one of these types of calls, for the rise calls, on the top for females and on the bottom for males. So this is on the third day after they've been presented with an opposite sex animal that has then been removed. And the groups are shown on the bottom. So these are the vehicle. These are the estradiol animals. Here's the lower dose, 0.5 mg per kg of Arachlor, 1221, and then here's the 1 mg per kg. And what you can see is actually this lower dose of Arachlor was the most strongly associated with large numbers of calls on that day. For some of the call types, we did see it with the higher dose of Arachlor, although not as much as this dose. Maybe there's some kind of inverted U-shaped dose response curve. But other types of calls, trill calls, had a similar kind of pattern. The flat calls, which are considered to be not hedonic, neither hedonic nor non-hedonic, they were unaffected by the treatment. So it actually is specific to these different call types. We also did some tests of other types of sociability and social novelty types of tests. So this test, which Michael Riley had run, and we just published this last year, has an EDC animal given a choice between different types of stimulus rats to look at, what is their preference for different social choices, males or females. They're also given the opportunity, in one case, to have a rat in one side but no rat in the other side to see if they prefer to be with a rat versus an empty side. And rats are very social, so you expect them to spend time with the rat, and they also tend to have a novelty preference. I can show you what these tracks look like here. So here's a case where we have an empty cage here, and we have an animal here, and you can see they spend a lot of time on the social side when they're running around. And then here's, if you give them a familiar versus a novel animal, you can see they spend more time with the novel animal. And what we found was that the EDC-treated animals, they weren't affected in this test, but they were affected in the social preference test, and they spent less time touching noses and sniffing and investigating the stimulus animal compared to the control animals. So this is another kind of social behavior that is affected. Third, we looked at a whole suite of genes that are associated with anxiety-like behavior. So this is the light-dark box. Rats, it's a big box about this big with a divider. One side is dark, just dim red light, and the other side is white light, and rats don't like white light. They prefer to be on the dark side. And so we can just put them in the box, track them, and see how much time and how quickly they move and what they're doing on the light side versus the dark side. You don't need to sweat the details too much, but just to orient you to the slide, the order of the treatments are always females on the left and males on the right. Control vehicle, estradiol, 0.5 mg per kg and 1 mg per kg Arachlor. So something you can see is, in general, females move faster than males. It's a sexually dimorphic behavior. But that with the Arachlor treatment, they actually, if anything, become a little bit more female-like in that behavior. Same with line crossings. When they cross that midline, Females do it more. That's really typical for rats. Female rats and female mice are more active than males. You can see the males are less active, but in this case, the higher dose of the Aerochlor, the males are more like the females. And so I'm not going to go through all of these different graphs, but this shows you that there's, again, a sex. In fact, I didn't talk about sex differences before for the other behaviors, but in this case, we have a sexually dimorphic behavior that is more apt to affect the males than the females. So I think looking at these behaviors is really informative, because now we can go back to the brains of these animals and determine what are the relationships between the changes we're seeing in gene and protein expression and these behavioral outcomes. We've also looked at a lot of different physiological and developmental outcomes, which I'm not going to go through in any detail. I'll just show you the body weight and the hormone data for corticosterone. But the birth outcomes and the sex ratios were not affected. We look at anogenital distance as an index of exposure to estrogens and androgens, and that was not affected either. But body weight was. What we looked at, so here females are on the top and males are on the bottom. And we've divided the body weight curves up into the prepubertal period through day 14 or day 21, during adolescence, and in adulthood, because the curves are so different. If you plot them all on a single graph, you can't see any differences. And what you can see, and it actually becomes more apparent as the animals get older, is that we do have body weight effects, which I think is really interesting, because our animals are only exposed to these low levels of EDCs for 2 days during gestation. And you start seeing these body weight curves differentiating out. These are the control males at the bottom, so they're the lightest. This dotted line here is the estradiol animals. This is, let's see, this is the 1 mg per kg Aerochlor group, and this is the 0.5 mg per kg Aerochlor group. And those are significantly different from each other, and we see a similar effect in the females in this case. And I was surprised by this finding because there really isn't a literature on PCBs and body weight, but there is a literature for some other EDCs showing that prenatal exposures to other kinds of compounds can result in body weight increases. And some people, as I showed you before, that curve of the chemicals going up and autism spectrum going up, well, obesity has gone up, including childhood obesity. And so maybe the chemical environment is contributing as obesogens. Sure.
Well, we don't expect the A12.1 dose to be higher in the body weight than the 0.5, or maybe I'm misinterpreting.
This one is the 0.5 and this is the 1. So we often see the dose response curves are complicated, and we don't have a full dose response design with only 2 doses. So I don't want to overinterpret what I'm seeing. But we have done a little bit more dose response work with that Aerochlor treatment. And what we often see is this inverted U-shaped dose response, where intermediate dosages will have a greater effect than higher or lower dosages. That's a really common observation both for natural hormone effects as well as for EDC effects. And there's a lot of mechanisms for why that may happen. For example, the lower doses may not be inactivating pathways that the higher doses may be inactivating. There are many endocrine systems that are being affected at the same time. But this 0.5 group actually tends to be exquisitely sensitive to a lot, or vulnerable at the level of the phenotypes that we're looking at. So we often see greater effects in this group than in this group.
So I have another question. Do you look for more perfusion tech?
Not in this study, but we're starting to do that now.
Good.
Yeah.
That's one explanation.
Yeah.
Yeah, because I guess the ghrelin and leptin hormones would also be partially controlled by the HPA.
That's right. That's right. Well, there's so much interaction between the hypothalamic control of energy balance and reproduction. Yeah, so we also have been saving serum from these animals, and so we've run some hormone assays, but we haven't run any of the metabolic hormone assays yet, but they're definitely on the list. And this body weight effect actually is incredibly consistent across our studies. And we're doing some transgenerational work looking at animals bred out multiple generations, and we still see the body weight phenotype actually heritable to the next generation. So I'm not totally sure what the explanations are for that, but presumably some sort of germ cell change that's happening.
Sorry, is that through the male or the female?
Yeah, that's a good question. So we actually see more of an effect through the females, but we see some effect through the males. Okay, and finally, last slide on this project. We also looked at serum corticosterone, and each one of these dots is an individual, just to give you a sense. There's a lot of variability, even in normal animals, for CORT. But you can see the 4 treatments. Females have higher CORT than males, another sex difference in the endocrine system. And again, the 0.5 group has higher baseline CORT than the other groups. Okay, so hopefully I've convinced you that these behaviors that are involved in reproduction, social behavior, communicative functions, and so on, are perturbed by prenatal EDC treatments during that very short period of development in a sexually dimorphic manner. Serum CORT is higher. We've looked at estradiol and testosterone, and those actually are not different. The body weight is increased in males and females, making me think that PCBs may be another one of these classes of obesogenic EDCs. I know I have to be out of here at 10 of. I was going to show you just a couple more slides, but maybe let me just skip to the end. And I was going to, this paper is published, so if anybody is interested, or I can maybe show this to the students at lunch. But let me just jump to the end so that maybe there's time for a couple more questions. Okay, so, I always feel like I have to go the extra mile to convince people that the work that we're doing in rats is pertinent to human health, which I suspect in this program at UCLA, it's not a hard sell. But when you talk to the public, I always like to point out that the conservation of these processes is incredibly high. People may challenge whether these behaviors are really good proxies for these kinds of behaviors in humans, but we know these behaviors are sexually dimorphic, and many of the differences we're seeing in our rats are consistent with the human epidemiological studies also showing sexually dimorphic differences in anxiety or autism or other kinds of behaviors, often in association with chemicals. We're hoping that some of the molecular work that we're doing may allow us to identify some targets that could potentially prove to be some sort of site for intervention. The timing of exposure is critical. I only talked about one exposure period, but there are so many critical periods throughout our life cycles, and it's not all prenatal. Pubertal, in adult, during aging, as our natural hormone environments are changing, we're going to be differentially vulnerable to these different kinds of exposures. And past contaminants, including PCBs, are a persistent and present problem. So just quickly acknowledge the NIEHS in particular for support. Lots and lots of undergraduates in the lab, graduate students are rats. They are happy. And this was our lab a few weeks ago. So I'll stop there and see maybe there's time for a couple more questions. So thank you very much. Questions? I saw one in the back first. No, we have. So we published one study looking at a mixture, a reconstituted mixture, which is thought to be a little bit more relevant to body burdens in humans. And I'm going to get the numbers wrong. I think it's PCB 153, 180 and 135. I'm going to have to confirm what the numbers were. And we saw, actually, kind of surprisingly to me, we saw actually a lot of parallelism with the Erichlor 1221 effects, but the effects weren't all identical. So we looked at a number of different endpoints, and we do see endocrine disruption effects with some, I should just say, with some overlap with the lightly chlorinated PCBs as well. Maureen, did you have a question?
I just was wondering, I saw that you were doing like EPN and other social related behaviors. Do you have a hypothesis as to which circuitries or systems maybe also?
Yeah. So I should disclosure, we actually didn't see any effects in the elevated plus maze. We only saw effects on the light dark box. So that might help us narrow down what the circuitry is, because those different tests of anxiety-like behaviors in rats probably aren't using identical circuits. So far we've looked at, we're only just starting to look at the brains. So we've so far just looked at corticotropin-releasing hormone, receptor, and BDNF, which are important in these behaviors, in amygdala. So, you know, this is tiny start to the experiment. And CRF receptor was significantly affected. BDNF was not. I mean, we're going to start to pick apart what the circuits are, but so I don't know the answer yet, but hopefully we'll get some insight into that soon. Okay, thank you.